I'm Smita Krishnaswamy. I'm from Yale University. Been coming to CGSI for a while. I think it's an awesome program. This year, Eleazar and company kindly let us do this two-part tutorial, which I would say maybe the first edition is given here. Other people in Australia and other places have asked us to deliver a similar one. So this is really trying to teach you guys about how to run a cellular dynamics inference pipeline on top of single cell data to derive cellular dynamics information rather than the static information that a lot of the standard pipelines derive for you. So I'm pretty sure you guys are aware of like Seurat and ScanPy. A lot of the operations in there just visualize the data, cluster it, differential expression, this kind of thing. But we think there's more information you can derive from single cell data. And it doesn't, in a way, depend on if there is many time points collected or not, just because we're collecting information about heterogeneous cellular states, and some of these are progressions of each other. We can go quite far if we really leverage the power of modern generative modeling in the style of diffusion models, but actually we predated that. I'll talk a little bit about that. So we can come up with continuous cellular trajectories, and that can be useful for a version of causal inference, which Sriram was talking about, but I'll talk about a different sort of timed version of causal inference. And I'm hoping we've gotten a lot of really good results from this, and I'll try to go over a small vignette where we've gotten results. We've gotten results in pancreatic cancer, stem cell biology, breast cancer, and I'm hoping you guys can do the same and also help us debug this pipeline. Zhao and Siddharth have put together an easy-to-use notebook, so check the notebook out at your own time, and they'll be around this week, next week to ask them. So that's really what it's about. It's a two-part tutorial. I'll give the first part today and the second part tomorrow. So again, it's called Deep Neural Network Models for Inference of Cellular Dynamics and Underlying Regulatory Networks. Really, the caveat that I didn't put is from single cell data. So that's really what it's about. So as you guys know, this is the general intro that I kind of give. A lot of our data in biology is very noisy, high dimensional, and sort of unlabeled. But we want to do a lot of interesting tasks from them. And initially, my lab was involved in doing the things that normal pipelines these days do, like denoising data. You may have heard of magic, or just visualizing data, UMAP, our method fate, things like that. But now we can really go beyond that. We can go further than that. And we can start to understand some of what the cells are doing, which is dynamic in nature. These are living entities. They change all the time. They alter their expression programs in response to different stimuli or natural conditions. And we really want to understand these as dynamic systems. And I would contend that with the data and the machine learning tools we have right now, we can actually start to do this. So let's talk a little bit more about this problem. Understanding dynamics from static snapshot data in the context of cellular populations can be something like, we have 3 or 4 time points. Can we interpolate what the intermediate time points were? For example, if you're studying embryoid body differentiation and you find that at the end there's these 2 completely differentiated or separated cell states, you might want to find out when did they actually split, because it doesn't look like they split here, but now they look split. And so you might not be able to catch that time with the coarse grainness that we have in terms of timing. Remember, in single cell data, the machine kills all the cells, so you cannot follow any cells in time. So we want to infer that continuous time. But another kind of inference that's very interesting is, if I just let this cell go under these conditions, what would happen to it in the future? Can I forecast it out? Can I generate its path? So this is cellular trajectory mapping, and all of these are done kind of in the same framework, so I'll talk to you about that. And what are the tools that I'm using? I'm actually going to go over the tools that are underlying this, even though you guys are going to get easy to click through notebooks to work on, to do this kind of analysis. Just, since you guys are maybe smarter than the average user, you can really look under the hood and see what's happening. So it's a combination really of data, geometry, dynamics learning put into a neural network. That's really what it is. So here's kind of our pipeline. And I'll tell you what I'm talking about today and a little bit of what I'm talking about tomorrow, and then what I'm not talking about, but you can come talk to me about it if you want to. So the idea is we have single cell data here, and we've collected it potentially at a few different time points, but not a whole lot, and there's gaps between the time point. So today I'll talk to you about how to learn these continuous cellular dynamics for every single cell. So we actually kind of pioneered this kind of idea way back in 2020 when the first flow models came out, continuous normalizing flows, neural ODEs, and that was called trajectory net. But since then we've moved on to something called MIOflow. And just a quick point of bragging for me is that the co-first author of MIOflow and the first author of Trajectory Net, Alex Tong, actually invented flow matching right after he left my lab, because he found out that for generative modeling you can do something like much easier and faster, but in biology you can't quite yet. So flow matching is really all the rage in image generation and generative modeling these days. Even Stable Diffusion uses it. And then so tomorrow I'll talk about once you have these trajectories, what can you do with them? It turns out that you can think about each gene causally upregulating, downregulating, or interacting with another gene over time. So you can do certain kinds of causality hypotheses generation using either something as simple as Granger causality right on top of these gene trajectories, or you could use something more complicated like a graph regulatory network that does a little bit more sophisticated version of that. That's what I'll be talking about. The last thing is, that's a little bit what we're aiming towards. We're aiming to see how do perturbations affect the system. So if you can simulate these perturbations via these kind of network programs, and then you can map out what all the different perturbations can do, then there's things we can do with it. And we have some methods that if you have experimental perturbations, you can map the perturbations nicely. We're trying to, so the last part is still work in progress, and if anyone wants to, whatever, work with us, chat with us about it, that's fine. So, but I won't be talking about that either of those days. So the outline is just, so I actually don't think I have anything on pre-processing, but Zhao will talk to you about pre-processing. But the notebooks, this is the order that the notebook goes in. The notebooks we give you will have pre-processing. Then I'll talk to you about embedding data with fate, which reveals the continuous trajectory. So if you don't have underlying continuous trajectories, or your embedding method lost them, then you can't do it. Then we'll talk about dynamic optimal transport, which is really the key mathematical idea that allows us to map trajectories in space. And then I'll talk a little bit about what you do after that. You can figure out, which cells are starting progressions. You can decode gene trends and things like that. So the first concept I'll talk about is manifold interpolating flows, or MIOflow, and its extensions. So I will say that the last published version of MIOflow was a few years ago in NeurIPS. Since then, we've extended it to tackle very specific biological problems in cancer, and you can find that in a preprint. We're also making what's called a journalized version of MIOflow. That's still in the works, but the first two you can have access to. The first is still published. Okay, so the MIOflow architecture roughly looks like this. It first embeds the data into a geometrically meaningful space. Within that space, the original MIOflow ran one ODE to determine how cells change instantaneously. This ODE network would give a time instantaneous derivative for each cell that you integrate out to give you the next state. Now we've added another one that's called a proliferation network, and it tells you how much the cell is that cell state is proliferating, so changing its mass or dying, so just kind of diminishing rather than being transported. And then the original MIOflow also used a multi-scale diffusion space, but since then we've found that the fate space is actually much better for cellular progressions. So again, in the examples I talked to you about, we do have these 4 or 5 different time points. If you don't, you can use something called diffusion pseudotime. And Zhao has a notebook in the GitHub repository. So if let's assume this was all one data point that was collected, but it still had these heterogeneous time points, you could run something called diffusion pseudotime to get different time points out of it and create a few different bins of pseudotime. And so what we do with these different points in time are we perform something called optimal transport. So literally thinking of this population of cells as some dirt that you push, but you're pushing it in a very efficient way. And as you push this dirt to match with the second time point, to match with the third time point, you are also pushing the grains of sand. So the mound of dirt is like your cellular population, and the grains of sand within it are your single cells. So you get a trajectory for every single cell this way, okay? And this is something very different than what RNA velocity or diffusion pseudotime or any of those do. This gives you a full trajectory for every single cell that you start with, okay? And sometimes that trajectory ends because we decide that cell died, but you get the full trajectory. Okay, and so here's the overall MIOflow architecture, and I'll go to it piece by piece. I'm going first with the fate regularized encoder and decoder. So what does this really mean? It's an encoder that puts cells from the gene expression space that you get. When you do single cell RNA sequencing, you get cells by genes. So that's the space it lives in. We're going to put it into the fate space. Okay, so why should we put it into the fate space, right? Well, first off, even deep neural networks, when they're reasoning about geometry, can't handle the amount of dimensions that single cell data has. It has 20,000 dimensions, and most of those dimensions are pretty sparse. So dimensionality reduction for denoising and other purposes is necessary. But what dimensionality do you reduce it to? So if you just put it through an autoencoder, you'll really have very little control over it. So you can look at some of the dominant types of dimensionality reduction. And what you find is some of the more popular ones like UMAP and TSNE can shatter trajectories very easily. So you want to put it in something that maintains continuity of trajectories where there is continuity. So something that preserves geometric distances. But not in the ambient space, but in a manifold reduced space. So that's why we use fate and took many years to develop and continue to improve it. The way generally as a snapshot fate works is it takes your cell by gene data, which you get in single cell RNA sequencing. It computes distances between rows of these, which are Euclidean distances. So that means for this cell, this has low Euclidean distance and this has far Euclidean distance. But then we invert those distances into a kernel where we only trust the close distances and we zero out everything that's far. So this gives a soft neighborhood for each cell. And the idea is that if you start a random walk from any of these cells, you only go somewhere in your neighborhood. And from there you go to the next place, and there you go to the next place. So you're creating a random walk through the space of the cells, and you cannot move outside the space of cells. And that's called manifold learning. You're like following the manifold of the cell. So from here, we turn these into diffusion probabilities for each cell to instantiate computational random walks on it and compute the probability that each cell will go to any other cell. So this restores global connectivity and an idea of how far every cell is from another cell, given that you can only walk through the cell space, right? It's different than this one. For example, these points are much closer because they have more paths going to them, right? And whereas here, they were all sort of equal. And then finally, this final step in fate is to take this new diffusion probability matrix and compute a new distance between its rows. And this is an information theoretic distance or a divergence. And this informational distance just embedded into low dimensions. So just to put it really succinctly, fate's just a method that preserves these informational distances that we call potential distances in low dimensional space. And so you could train an autoencoder to do this too. You can compute these low dimensional distances on your training data and then say preserve them in your space. So that's kind of what we do. Just to go over some of the ideas of it, you might have also read about these ideas in our MAGIC paper, several other papers. The Euclidean distances that you compute in the second step will give you a distance between every pair. And it's not really clarifying the shape of the data. But when you kernelize it or put these distances pointwise through a kernel, you start to create paths that follow the shape of the data. And then the diffusion step, as you power that operator, you're diffusing T steps. And so the way you get from here to here is this is an affinity matrix. Row normalizing it will give you a diffusion operator, and then powering it will give you probabilities of T-step paths. So powering it higher and higher makes it conform more and more to the low manifold dimensions. So what is the cool thing about fate is, first off, we denoise the connectivity to go through the data, through the diffusion. We've re-represented the cells by their t-step diffusion paths to every other cell, so it globally contextualizes them. Then the potential distance is just embedded through MDS. So this is the potential distance. It's the distance between 2 points or 2 cells. It's just Euclidean distances between rows of the diffusion operator after log scaling them. Turns out that this log scaling is important for making it a divergence and considering far distances as well as near ones. And here's the comparison on the same data set. These are embryoid bodies differentiating through different precursor lineages, progenitors of cardiac, neural, and hematopoietic. That's what you kind of see, cardiac, hematopoietic. These are neuronal lineages. And you see that Fate does a better job of figuring out what these, or showing what these trajectories are and how the differentiation paths are going away. And so we developed Fate with this sort of in mind. So to regularize autoencoder for Fate, we just take an encoder and decoder framework. You can start from PCA dimensions here, which is kind of a pre-reduction into a larger space. And then within this latent space, we add a loss to preserve potential distances. So we're saying the position of any cell from any other cell in this latent space. So this is a pairwise loss. You go through 2 cells at a time. So this distance, the Euclidean distance in this latent space, we're penalizing it from being different than the potential distance. That's the Fate regularized autoencoder. The other use of this is you can back out of it. Once I put a point into regular Fate, it's very hard to back out. I can't move in that space and just back out. It doesn't have a decoder, right? So now it's like PCA. It has a decoder and an encoder. You've encoded and put stuff into Fate space. You can move through there. You can create trajectories, and you can decode any point you want back into the gene space. So that's the other advantage of it. It's extensible, so you can add new points, take new points out. Okay, so now the flow network. This is a derivative network. So this is a neural network that generates time instantaneous derivatives parameterized by time. So time 0 derivatives are derivatives on the data you measured, and then you could have another time point that's even in the gap between the data set. So this derivative network is accessible to an ODE solver. So when you ask it to solve, meaning, okay, give me the state of these cells at time t1, it can query the ODE, it can query this derivative network repeatedly, however it wants. But we let the ODE solver do that. So the ODEs, they're fancy ODE solvers that sample more where there's curvature. There's simple ODE solvers like Newton's method that sample consistently. And how is this penalized to be self-supervised? I talked about this data being unlabeled. We just, the main penalization is that if we have another time point at which we actually measured the data, it matched the marginal at that time. That's the main penalization. But let's talk a little bit about how this can be modified a little bit to give what's called Earth mover's distance or optimal transport. Again, as I said, Earth mover's distance is how you move one distribution that you consider as a pile of dirt or earth into another one. But it's not just about moving it in any which way. It's about moving it in the most optimal way so that you're doing the least amount of work. So what it actually is if you have these 2 marginals and you're transporting mass from each bin to another bin so that you're doing the least amount of work. So this joint probability distribution here defines a transport plan. The cost of this transport plan is defined by this distance matrix here. And actually this would be a linear program where you minimize the cost subject to the conditions that this marginal match up with distribution A and this marginal match up with distribution B. So we don't actually have to do this. We can get our neural network to do it without running any linear programs. Linear programs itself an optimization. And plus we can do one more thing, which is give us dynamic optimal transport. So let me tell you what dynamic optimal transport is. Static optimal transport is very happy if you just match these cells with those cells, but it's not really specifying the plan in between. So you could have squiggly paths or whatever paths you want. Dynamic optimal transport also controls the path length here, and we do that by penalizing the magnitude of the derivative. And this gives us smooth and more plausible cellular trajectories, because we don't have this amount of high frequency information to infer these squiggly trajectories. So we're inferring the simplest trajectories possible from that. And we've proven in the paper that if you regularize it this way, if you penalize this way, this indeed gives dynamic optimal transport on your ground manifold space, so in the phase space. Another thing, so this is really important. So let me emphasize why it has to be in the manifold space. So if you have that population, you would not want a trajectory inferred that looks like that. The reason is because that doesn't seem like a plausible path. We didn't find any cells along that path. It doesn't seem like viable cells live in that path. You want to follow the cellular manifold. So another way we enhance its behavior to go on a walk exactly in the manifold is to penalize paths where the neighbors are far. So if you're far from the nearest cell, then we're going to give a penalty. So it's again emphasizing that where you have density, you would go through the density of the data. Okay, so cell growth and death. The original MIOflow didn't really have this. The original trajectory net had a version of it, a version that I find slightly hacky. It used static optimal transport to see these growth and death rates, and then it would use those. But the new enhancement of MIOflow that is available also has another neural network that for each cell talks about what its new mass is going to be. So is it going to, instead of these cells, instead of being forced to do this transport, they would just die. So they would have low mass, so mass less than 1. So multiplicative mass less than 1. And the cells that are growing, like these cells, would have mass more than 1 because they're proliferating. So that makes the system more realistic. It's not pure optimal transport. There's also cell growth and death. And so this is actually why we still need the ODE, and we can't take the shortcuts of generative modeling because we need this rich behavior here. Okay, and finally, this is what the dynamic optimal transport sort of looks like. I'm not sure what version of it this is, but I've asked Zhao to give me a brand new version of it. But again, pushing the mass and leaving trajectories in their wake. So what can you do with these trajectories? So first off, you can look, as I told you, these are different fates that the cells end up in. How did they all get there? They all started as human embryonic stem cells. And, all of us, we all started as one egg, right? Or something like that. And we have all these different cell types now. And that's really what these developmental biologists are trying to figure out. And you can see, if you decode back to the gene dimensions, you indeed see that their gene trends are different over the developmental time. So the bottom is the developmental time, is not wall time. And you see how different genes develop differently. Some genes go up and some genes go down. They can have very non-monotonic behavior to specify the cell fate. And a kind of analysis that we're really excited about doing, particularly not so much in stem cell development, because the cell of origin is semi-obvious in stem cells, but more in cancer. And I might go through a quick cancer example. Is we can also order, do I have that here? Oh, I have it later. The cell of origin analysis looks something like this. At day 0, where do you start your trajectories? Some of the trajectories dropped, some of the cells are dropped, and a lot of the cells start here. So you can do something called the cell of origin analysis. So after you do the cell of origin, you have, you can also figure out how cells get from that origin to your cell fate specification. So here, what we're doing is we're ordering genes by peak time, like when they come up. So what you see is that here are the genes that come up first, here's the genes that come up next, here are the genes that come up later, and you can have a full time trend for each of these genes in each of these trajectories. So you have these different trajectories here that go to different time points. You can group the trajectories and you can understand that. And finally, I will not talk about this today. These continuous time trends definitely enable less correlational, more causal analysis because, for example, you can perform Granger causality. You can say this gene goes up first, and from it you can predict the value of the next gene. So this gene might be Granger causing that gene, which is a hypothesis generation method. You of course have to validate it. Here's a quick application to breast cancer data, which we're about to submit somewhere. We had to redo this whole story since I talked about it here last year. So it's taken us a bit of time. These are breast cancer organoids or mammospheres grown over 30 days. You see they're seeded as single cells in 2D culture and they grow into these 3D organoids. We have 5 time points of data. So these 5 time points of data, we're sampling these at different points in time and measuring them. So you seed a lot of them, and then you take out some at each point in time. And then you see, by day 20 and stuff, these mammospheres are really big. Then we're doing some assessment of what the cell states are. These cells are very epithelial. These cells are somewhere in between. We don't see any super mesenchymal lineages here. And we also see that some cell types are dying. And then when we run our MIOflow, and we're calling some of the enhancements biologically inspired enhanced MIOflow, we see that there's 2 trajectories that emerge. This looks like it's a tumor-forming trajectory, and this looks like this eventually goes to apoptosis. So we become really interested in the tumor-forming trajectories. That's where we've performed the cell of origin. Where do these 2, so we seeded a bunch of cells. Most of them died off. What were the cells that actually gave rise to these tumors? So we can do the cell of origin analysis at day 0, meaning where did these trajectories all start? They start from this small area. We can characterize these, and this can be considered for this cancer an enhanced cancer stem cell signature. So we've actually experimentally validated that if you have EpCAM high, CAV1 high, and actually some kind of cell cycle specificity using sorting, that these are more likely to create more tumor spheres in the well. So here we have these trajectories over time. We can compare the 2 types of trajectories to see how the gene dynamics are different. We can get these timed gene expression modules that then we used Granger causality with. So the Granger causality is limited. So we have these 1, 2, 3, 4, 5 gene expression modules based on when these genes peaked. So we only let genes in the earlier peak cause the ones in the later peak. So that gives you a time ordering of which way the directionality goes. So once we do that, we use this Granger causality analysis to look at what genes could be Granger causing one another. So now the real way we used this was we knew there were a bunch of genes implicated in this cancer. This is triple negative breast cancer. There's very few targeted treatments. We wanted to see what genes were upstream of it or downstream of it, new genes that people hadn't really considered that much before. So we took these new genes out of the Granger causality plot and came up with a bunch of ones. And one of the ones we kept seeing that was a huge hub for everything was Ezra. And at this point, we've done a lot of mouse model validations. I haven't done it myself. My partner, Christine Schaefer in the Garvin Institute did it. So you can tell that if you use Ezra with the vehicle, you get the best tumor response in these mice that have xenografted triple negative breast cancer data. And you can also see the normalized radiance when you measure it with flow cytometry for a metastatic site. So this is kind of where we are. So a lot of the tools from my lab are on my GitHub, and my GitHub is here. Zhao, are you going to very quickly show where the notebooks are? Or you can do it in I have it up. So Zhao has prepped this notebook called the Omics Toolbox with this pipeline. Yeah. So I don't know if you need my mic.
No, I'll be quick. So the whole idea is, because we have a bunch of tools and different tools, we decided to use everything on notebooks. So I know we were discussing recently, we should have just command line tools, but in our case, we're always using Python notebooks. So it's basically if you guys go under notebooks and MIOflow, in this one. So you have all the steps to run everything. In this case, if you want to run the actual MIOflow, you can go to the MIOflow package itself. This is just to show you step by step. So for example, if you go to the actual
maybe
MIOflow,
click into the Fate lens.
Yeah. So the first one is just set up, and you just read through it and download the embryonic data. For example, if you have your own data, you just follow kind of the same steps. And then here is it's a bunch of things written and explaining what you're doing, but the whole operation itself is pretty simple, right? You just instantiate the operator, and then you feed it, and then you get the outputs, right, from it. So you can just kind of plug your data in and try to see everything. An example here is, for example, the autoencoder. I think the output will be bad at GitHub. Yeah. But basically, this is just showing you, it gives you details on how you train this autoencoder, because if you go to the actual MIOflow package, this will be hidden from you, right? So the idea of the notebooks here is just to show like step by step. And a classic problem we usually have with single cell data is you don't have time measurements. So embryonic data, we have the time measurements. In a lot of cases, we don't. So we explain here so this in our case, we do have the time measurements. But if you don't, we have like an explanation here on how to impute a time feature. And it's using diffusion pseudotime, which is a different paper and a different method. It's from a collaborator, but it's not exactly from our lab. And then basically it tries to explain you how to do it. Like you create a root cell and then you diffuse through time there and you get some kind of like, it's kind of like the walks Smita just showed.
It uses diffusion also.
Yeah, so in this case, you see in our data, we know we have like 3 different endpoints. But if we didn't know, like this would be the endpoint, right? Because you're just walking on your manifold. So you need to play around a little bit for your data type. If you know you have a final, like exactly final output, this is really good. So now you have a time measurement for each of your cells, and you can do optimal transport on top. But yeah, so we can talk about it later because I think we're kind of out of time. But yeah, and feel free to give me feedback. Like this is like all under construction, so it's good if you guys are just like trying to run, and on the first one it's breaking. Please let me know.
Maybe show the visualization.
Oh yeah, sure. So yeah.
What they would get at the end of it. You'll get the similar trajectories like I was showing you.
Yeah, you get the trajectories, and then you can, of course, like play around with them, like just visualize some of the trajectories. Like this would be an expected one or a different one.
You see like a bad one there, you can filter.
Yeah, you can filter. And then here it's explaining how to decode back to gene space. So from those trajectories, oh, there's a visualization. We removed this one, but it's just visualizing in PCA space. But basically, how do like the trajectories look back in gene space? And this is an average of all trajectories. So we can see, like for example, this one has like really high variance. So this trajectory is not really something you found. And then this final one, you have the heat map where you just organize all your genes through time, and then basically you can start inferring like this range of causality that Smita will talk more tomorrow. Yeah, so if you guys feel free to play around, it's on the lab GitHub, it's on Mix Toolbox, and of course the tools itself have their own tutorials, but they're more like you just use it, right?
So under Omics Toolbox, it's under notebooks.
Yeah.
So the others are other packages.
Yeah. I think that's
All right. Well, thank you guys.